everyone, I hope you're all doing well. Today we're going to be talking about the anatomy of the eukaryotic cell. So this includes plant cells and animal cells, and I don't think there needs much more of an introduction, so let's just get started. Alright, so we just have one slide today, and this is it, and it's basically just a very labeled diagram of a eukaryotic cell. I'm going to be mainly just going through the compartments one by one, and you can just read along with me if you would like. So, starting at the top right, we have the lysome, which is a membrane-bound organelle containing hydrolytic enzymes. And this is gonna degrade absorbed substances or micromolecules that have to be recycled. So it's like almost a trash bin. It's gonna help with the unwanted molecules of the cell. So going down from that is the vacuole, which is a big vesicule. It helps with digestion, waste storage, and evacuation. It also holds water in plant cells. It's very important in plant cells. So the vacuole and the lysome are very useful for getting rid of waste. Now, going down again, the peroxisome is another type of waste management, except this is more of a metabolic compartment with a simple membrane, and this has an extremely important job, which is to transform oxygen to dioxygen. And oxygen is actually really toxic in the cell because it's very electronegative, so it wants to hog everybody's electrons, which is not good. We want to keep their electrons where they are for the most part, so it's going to transform oxygen to dioxygen, and with that we can form water with use of other enzymes. So I've put in the reaction for you there. It usually transforms hydrogen peroxide into two molecules of H2O and dioxygen. Hydrogen peroxide is the reason it's called the peroxisome. Unlike the mitochondria and the chloroplast, as we're going to see later, it does not have its own DNA or ribosomes. Next, the one that you probably know the most about, it's the nucleus, which holds all the genetic information. It is surrounded with the nuclear envelope, which is pierced with thousands of nuclear pores. So it contains the chromosomes and the nucleoli that participate in the synthesis of ribosomal subunits. In direct contact with the nucleus is endoplasmic reticulum. So I wrote it once ER and RE. That's because in French we called it réticulum endoplasmique instead of the other way around, but people will understand that you mean the same thing. There are two types, the smooth and the rough endoplasmic reticulum. Let's start with the function of the smooth. The smooth endoplasmic reticulum is going to synthesize lipids, carbohydrates, and is also going to stock calcium ion and helps detox medicine and alcohol. So if you're kind of seeing a trend here, there's a lot of compartments that help the cell stay safe from external products. The rough endoplasmic reticulum, which is in direct contact with the nucleus, helps with protein synthesis and secretion of proteins involved with ribosomes. It also produces glycoproteins with carbohydrates and assures membrane growth. So these two, the smooth and the rough endoplasmic reticulum, are a continuous membrane system. Next up, another really important compartment is the Golgi apparatus. It is formed by packed membrane enclosed discs that we're going to call cisterna that have a polarity, so plus and minus. You can kind of see it as a dispatch station of protein products that were received from the ER. So it's kind of like the post office of the cell. Some people like to call it that. I told you it has a polarity. The cis end is going to be for receiving and the trans is going to be for shipping. Now for the two that you are probably most familiar with, which is the chloroplast and the mitochondria. So the chloroplast is an organelle isolated from the cytosol with a membrane that contains the thylakoids, which helps with photosynthesis. So obviously the chloroplasts are only going to be in plant cells, not in animal cells. Mitochondria that are in both, they are surrounded by a double membrane and the inner part folds inwards, and they help with cellular respiration. So these two are metabolic compartments. and they have each their own DNA genome, which is double-stranded and circular, and they have their own ribosomes that are of bacterial type. You're probably wondering, first of all, why do they have their own genome? And secondly, why are they of bacterial type, being circular and all? And that is because of the way eukaryotic cells actually first appeared, and it was in symbiosis with these little organites. Mitochondria and chloroplasts used to kind of live on their own until they integrated into these cells, and they were kind of helping each other out. So they formed a little alliance, and so they kept their own DNA. So the cell has its own DNA, and the chloroplast and mitochondria have their own DNA. And these two were prokaryotes. Last but definitely not least is the ribosomes. Those are kind of everywhere in the cell, and they are produced in the nucleoli, which is an inner part of the nucleus. The ribosome is 2 units composed of proteins as well as ribosomal DNA, and they help with protein synthesis. So I know I didn't go into a lot of detail about the function of these compartments. I kind of wanted to give you a bit of an overview, but many of these compartments we can definitely go into detail into further chapters. I'm thinking photosynthesis, I'm thinking cellular respiration, protein synthesis, whatever you'd like. So, as always, I hope this was helpful. If you have any questions, please leave a comment below. If you have any suggestions, I would love to hear them as well. And if you would like me to go over a specific topic, whether it be in chemistry, physics, math, or biology, I would love to do that as well. As always, stay safe and happy learning.